So welcome everybody to the October 14th seminar of the IMAG MSM Working Group on Multiscale Modeling of Viral Pandemics. I appreciate the speakers for being willing to come and talk to us. They've generously agreed to stay on afterwards if there's questions and discussion. I think after last week's presentations, today will be, hope we'll have some lively conversation about the material. I need to remind everybody that the meeting is being live streamed on YouTube and that those recordings are made available publicly. So please be aware of that as we go forward. So as always, you have myself, James Glazier, Reinhard Laubenbacher, University of Florida, Bruce Shapiro, and Jim Sluka, who is on holiday this week. We are always available to take suggestions, comments, complaints, and anything that you would like to do so that we can make this group more effective. We have our usual social media. We have the IMAG MSM wiki page. There is an IMAG LinkedIn page. We have an IMAG, YouTube channel. And we also have a Twitter account and a Slack channel. And we always appreciate it if you help us populate those and make sure that we are productive. I think in particular that we've had a smaller turnout the past week or two for some outstanding speakers. And so any ideas people have about ways to advertise these meetings so that people understand the importance of the content that's being generated would be much appreciated. Are there any announcements for today before we get started with the workshop? No. Okay. All right. In that case, upcoming, we have a quite good productive schedule coming up. Next week on October 21st, we have Jason Shoemaker and Tiam Hedari from UBC talking about, in the one case, modeling, in the other case, computational analysis of RNA-seq data. October 28th, we have a talk on robotics in biomedicine, and another talk on SARS-CoV-2. November 4th, we'll hear about malaria, and then on testing and its role in controlling infection. So we have a great set of talks lined up for the next couple of weeks. If people have suggestions for speakers and or want to speak themselves, you're most welcome to put yourselves forward or suggest names, and we'll get them on the invite list. So we always appreciate your help in generating our list. If you are a subgroup leader and you would like to present the work that you've been doing in the subgroup, or if you have an idea where you'd like to have an open conversation about a topic that you think is important, that's fine too. We don't have to have use these slots only for the purpose of seminars. So please remember to mute your mic if you're not speaking. If you see somebody Zoom bomb us again, I appreciate your help in letting us know about that so that we can control that appropriately. And without more further ado, we will go to our first speaker, Balazs Madas from Budapest. As you know, I don't do an elaborate introduction, so to save time, we will turn it over directly to Balazs.
Thank you very much. And thank you very much for the invitation. And I hope I can share the screen. Yeah. Do you see my slide in full screen? Yeah, great. Thank you. Then I hide you to see my slides. So thank you very much again for the invitation. I first have two comments. You see here that there are 7 authors of this paper, which was published at the end of the last year. And I was asked to have a rather high-level presentation, but the problem that the people who did the modeling are not me, and it's rather late in Hungary, so I wanted to invite one of my colleagues who probably could better answer any questions than me, but yeah, so they were not very happy to or I asked someone and he preferred not to come, but we are ready to answer any questions if they come. Yeah, later, you know my email address, or if not, I can share it later. So if there are any questions which I can't answer, I will try to do so later to find an answer for that. And the other comment, so this study was published on the last day of 2020. You can see the link here, or you can directly take, if you cannot click on the link, you can take a picture from the QR code and you can already check it if you want. And what I think it's interesting regarding this COVID-19 related research, that if you look at the paper, which is 10 months old, you may feel that it's quite outdated. Actually, when we wrote this paper in May 2020, and I cited the paper, which was 3 months old at that time, then I already had some bad feelings that maybe it's completely outdated, and in the data which I take out, maybe there are more relevant data since then. So I apologize if something is outdated. Actually, it's not our main research. We are not a group of related to infectious diseases. We mostly work radiation protection related topics. However, now we also have a small project started in September, dealing with this SARS-CoV-2. So any comments that something is outdated is very welcome, because I have to catch up the situation. So sorry for this long introduction. So yeah, so the early scientific response to COVID-19 had 2 focuses. One focused on the understanding of the spread of the disease, which means epidemiology, and the other part was focusing on pathogenesis following cellular entry, which we may call physiology or cell biology. But the feeling was that much less attention was paid to how this virus from the environment reached the receptors of the target cells in the respiratory system. And it also seemed important because these receptors are available all over the respiratory system. So the transport of the virions seems to be important, especially because at the beginning it was already clear that there are different kind of symptoms. There are mild upper respiratory symptoms, and in some cases there is severe pneumonia, and it seemed important that the outcome of the disease can be determined which cells, or where are those cells which are infected by this virus. And it was also clear rather early that that was the paper which seemed to be old at that time, that the viral transport towards the deeper airways has to happen through the respiratory system and not through the circulatory system, because viral RNA was not detected during the early phase of the disease in the blood. So the aim of the study was to characterize the deposition of distribution of SARS-CoV-2 in the airways upon exposure to cough-generated droplets and aerosol particles. Of course, it is just one scenario, but that seems to be an important scenario. And for this purpose, we applied the Stochastic Lung Deposition Model. Here you can find some references and you can check. There it's a rather old one, so it's more than 30 years old, the first publications which came out, but it was extended and developed since then. And the model itself, so it, of course, the different parts of the respiratory system, and there are different methods for calculating particle deposition. In the extrathoracic airways, it is computed by empirical formulae based on measurements in hollow airway casts, and which is more relevant for this model that in the intrathoracic airways, particle deposition is simulated by reconstructing the paths of inhaled particles in a stochastic lung structure. So what does a stochastic lung structure means? That there are databases of the airways containing the airway lengths, diameters, branching angles, and gravity angles. And if we know the distribution of these data in different airway generations, then we can select a path randomly using these distributions, and here we can follow a particle path. So these particles are tracked from inhalation until they either deposit or leave the airways by exhalation. So both the inhalation and the exhalation process is modeled. And there are different mechanisms by which particles can deposit, and impaction, sedimentation, diffusion mechanisms are all considered. And of course, there is very important what kind of input data we use. And here we use the functional residual capacity, inhalation parameters like the inhaled volume, inhalation time, breath hold time between inhalation and exhalation time, and breath hold after exhalation. And of course, which is also very important, the particle properties. Regarding the inhalation parameters, we suppose normal breathing in sitting position. And for the particle properties, we used an aerosol size distribution, which were taken from a publication describing the size distribution of aerosol particles from coughing of influenza patients. And of course, it's a different disease, but there are evidence that the different respiratory infections cough from different respiratory infections cause very similar size distribution. So probably, or surely there are newer data since then, but at that time we were using this size distribution data, which you can show in this figure. And using this input with the normal breathing mode, you get deposition distribution. And what we first found, that if you look at the mass deposition fraction, so which ratio of the inhaled material is deposited in different parts of the airways, you can see then more than 60% deposit in the extrathoracalis airways, around 5% in the bronchial airways, and 8.5% in the acinar airways. If you look at another, if you look at the distribution over large, small, and peripheral airways, then we see the distribution here. So if we look in the intrathoracalis airways, and most of the inhaled mass deposits in the peripheral and in the small airways, and somewhat smaller quantity in the large airways. So if we look only on these data, then we see that this cough-generated particles reach all parts of the airways. Of course, much more in the extrathoracalis airways. It's not surprising, that is a function of that, but everything is reached. However, if we look, this is another part which also shows that the same, that everything is affected, but this Stochastic Lung Deposition Model is can determine the deposition distribution over different airway generations. And here you can see that the most affected part in terms of deposited mass is the 19th and 20th airway generation. So if most of the viruses which penetrate the extrathoracic airways passes 19 bifurcations before depositing. And what is, of course, very important, that it's the deposited mass. So if we would look at the deposition density, then you wouldn't see such a peak, because the area of the 20th generation is, of course, much higher than the airway generations before. So, but we can see that the inhaled mass reach every part of the airways. However, if we try to estimate the viral loads or the infectibility or the potential infection potential of the inhaled material, then we can make the following calculations. So there were data, and there are, of course, a lot of data on viral loads in the throat swab samples, which can be a source of the cough-generated material. And of course, it is a very wide range. But so if we consider the median value, then we get that we need around 2,500 breathing cycles to result in one deposited copy of RNA in the acinar airways. So it means that it's a very low probability. And even if we consider the maximum values, then we still see a very low 4,900 copies of RNA in the extrathoracic airways and 680 copies of RNA deposit in the acinar airways from a single inhalation. However, if we consider the difference between the viral load in terms of RNA copies per gram and infectibility, which means that a minimum viral load of a sample to be infectious is about 1 million RNA copy per cubic centimeter, then the probability of direct acinar airway infection from SARS-CoV-2 inhalation of cough-generated droplets and aerosol particles is very, very low. So that was which was surprising for me at that time, that even if we inhale directly cough-generated material from someone who is already has this COVID-19 disease, the probability that virions reach your acinar airways and you got infected, it's very, very low. So what does it mean? The main what we can find from that the first step to get COVID-19 is that you get an upper airway infection, and this will later develop into pneumonia. Actually, it is in agreement with the clinical observations that first symptoms of COVID-19 were related to upper airway infections like cough, anosmia, ageusia, and the clinical deterioration happened after around 1 week. And this finding was also in agreement with the virological analysis of mild cases who had very high concentration of viral RNA in the throat swab samples, and the virus itself could be isolated also from there. It showed that there is an active replication in the upper airways. So this suggests all this, that first your upper airways get infected, and then the viral load increases there, and there will be a huge amount of SARS-CoV-2 in your upper airways, and later this will develop into a pneumonia. Of course, what is needed for that is that the viruses from the upper airways go down to the acinar airways, and yeah, and what can be the way for that? So from the throat of patients with mild symptoms, viruses can be transported either via the respiratory system or the circulatory system, but there were evidence that viral RNA couldn't be found in the circulatory system in patients with mild cases. So that shows that that viruses are transported via the respiratory system. So the potential mechanisms include the reinhalation of our own cough, aerosol and droplet generation in the throat during inhalation, virus transport on the surface of the bronchial airways, or gradual infection of neighboring cells expressing ACE2 towards the periphery. And it also shows that compromised mucus production on transport can be an important risk factor for COVID-19 pneumonia. This may be some diseases affecting the mucus production and transport can also increase the probability of severe COVID-19 in this way related to mucus production or transport. And what is an important example for prolonged exposure, which of course helps or increase the probability of acinar airway infection, is the continuous reinhalation of own coughs of infected patients, which may contribute to the progression of the disease. So reducing our reinhalation of own coughs could significantly prolong or even block the onset of further more severe phases of COVID-19. So using a tissue or cloth in order to absorb droplets and aerosol particles emitted by out coughs of infected patients before reinhalation is highly recommended, even if they are alone in quarantine. So that was one finding of our work. And the 1 week difference between the onset of initial mild symptoms and clinical deterioration provides a precious window for prevention of pneumonia. So any way of blocking or significantly reducing the transport of the virus towards acinar airways can be very helpful. So, and the suggestion at that time was that disinfection of the mucosa of the upper airways may help to avoid or prolong the progression of the disease. Yep, so that is the end. So thank you very much for your attention. And if you have any questions, you can ask, as I know, later after the second talk, but it's up to the organizer. So thank you very much.
So thanks for inviting me to present our work. I heard that you discussed booster doses and our work in previous week. So I thought that what I'll do is I'll share with you some of the updates on the data that we've published previously. So I'm going to present today an update on the analysis that was already published in the New England Journal of Medicine, but the data that I'm going to show you here is also publicly available now on medRxiv, and I'll have a link inside the presentation. So if anyone is interested in the details, they can go and look at them. And I'll also say that this work was done in collaboration between 4 or 5 different research institutes in Israel in collaboration with the Ministry of Health. So there's many people involved in this work, but I'm going to share the summary of the results. So before we start talking about the results, I just wanted to share with you some insights for why I think Israel is an interesting country for studying the effect of vaccines and the vaccine waning and the effect of boosters. And the main reason for that is that Israel had a very swift vaccination campaign in the beginning of 2021, and that resulted in the fact that in Israel we had a very large proportion of the population vaccinated very early on. So in fact, we are ahead of most of the countries in the world by around 3 to 4 months. And in this sense, Israel provides some kind of a window for the future for other the situation that's going to be in other countries. So we hope that by sharing the data that we collect in Israel, we can help other countries better prepare for the future. And beyond the fact that Israel had a very early vaccination campaign, two other important facts about Israel is that we have a very large testing capacity. So I think we are like in the top 10 countries in the world in terms of testing capacity. And we have very comprehensive electronic medical records. Israel has centralized the healthcare system, and it has been digitized almost fully for more than 2 decades. So we have very good framework for following up on cases. So as I told you, in the beginning of 2021, Israel had a very swift vaccination campaign, which resulted in a very strong decrease in the number of cases that were found every day. But from around mid-June, we saw a resurgence in cases, mostly due to the emergence of the Delta variant and a combination of the Delta variant with the waning of immunity. And that resurgence has not just been expressed in terms of the number of cases that we have, but also in the number of severe patients that we have, and a striking figure about the number of severe patients that we had in the end of August is that the majority of them were fully vaccinated, which suggested to us the fact that the vaccines are waning not just in terms of their protection against infection, but also in terms of their protection against severe disease. And during this time, the end of July and the beginning of August, we had projections that the number of severe cases is going to increase exponentially and going to cross the capacity of the healthcare system to deal with the severe cases. That resulted in the country deciding to administer booster doses, initially for the elder population, for people above 60, but then gradually this campaign of booster vaccinations has been extended to other age groups. And now in Israel, we have boosters open for all age groups above 16 for people who got 2 vaccine doses and more than 5 months have passed since the second dose. As you see, the uptake of these booster doses has been pretty substantial. We have more than 3.7 million booster doses administered out of around 5.5 million people who got the second dose. So as you can see, for example, for the 60 and above population, we've reached more than 90% of the eligible population getting the booster dose. So this prompted us to try and look at the effect of the booster dose on different outcomes. And for that, we collected the data from a centralized database of the Ministry of Health, containing data on about 4.6 million people who were eligible for the booster dose. And this data covers the period from between August to October. And we have in it more than 100,000 confirmed infections, and more than 1,000 severe illnesses, and more than 250 deaths. So it's kind of a rich data set for us to start and look at the effects of the booster dose. So I think that you've probably seen this a version of this figure in the previous week. This figure is a summary of our results showing the dynamics of the protection from the booster dose as a function of the days since the vaccination with the booster dose. What you can see here on the x-axis is the days since getting the booster dose, going all the way from 0 to 58. And on the y-axis, what you can see is the fold reduction in the incidence of confirmed infection between people who got the booster dose compared to people who were eligible for the booster dose, meaning that they got 2 vaccinations and more than 5 months have passed since the second vaccination, but they didn't get the booster dose. So we are basically comparing here people who got the booster dose with people who didn't get the booster dose. In order to do this analysis, what we use is Poisson regression. We are trying to model the rate of confirmed infection in this case between the group of people that got the booster dose and group of people that didn't get the booster dose. And what you can see here is the rate ratio between the groups. So, for example, in day 16 following vaccination, we see that people who got the booster dose are about 10-fold less likely to be confirmed as to have confirmed infection compared to people who didn't get the booster dose. And what you can see here is, if you remember from the previous week, we saw this pattern that in the beginning we had a protection, which is a kind of it's not a true protection, it's kind of an artifact from the fact that people that get the vaccination usually don't get tested, or there's a selection for people who weren't infected to get vaccinated. So in the beginning, we see some kind of a protection that is an artifact from the fact that people are not getting tested in the couple of days after vaccination. Then we see this transient artifact disappearing. We have a period in which we don't have protection because the boosters are not kicking in yet. And then after about 12 days, we start to see the boosters get in effect. And the number of 12 days makes sense from an immunological perspective, because it takes about several days for the immunity to build up after the booster doses. And it takes also another couple of days between when the biological effects kicks in until we see it in the data, because people who get infected, it takes them several days to develop symptoms because of the incubation period and to go and get tested. So we expect this effect to start appearing about 12 days after vaccination. This is what we see. The data here is similar to the data that we had in the New England Journal paper, but it now extends to a longer follow-up period. So as I told you, this data is already published in the medRxiv. You can go and look it up if you're interested. What we can do with this new data is to extend the age groups that we're looking at beyond the elderly and look at other age groups. Sorry, I didn't tell you what is the final result from this. What we can do then is we can take all of the people after 12 days, so 12 days and more, and look at the number of infections that they got and compare it with the number of infections that people who didn't get the booster dose experienced. And we use the number of infections relative to the number of days at risk that people experience during this period. So for example, for the booster dose, we got about 2,700 infections. from around 46 million person days at risk. And for the non-booster dose, we got 12,000 infections from around 20 million person days at risk. And we take these two numbers and we put them into the Poisson regression, taking into account all of the covariates that can influence the rate of infections. For example, the rate, the gender, the demographic group, the period at which people got vaccinated, or the incidence rate at the location of residence of the people. We get an estimate of around 12.4, meaning that the rate of infection in people that got the booster dose is about 12-fold lower than the infection rate in people who didn't get the booster dose. So now we can do it for other age groups. So you can see here, it's sorry for it being small, but you can see a similar figure as I showed you before, but now also for 50, 40, 30, and below. As you can see, our follow-up time is different for different groups because of the fact that the administration of the booster doses started in different time points. It initially started in people at the age of 60 and above, and then got extended to younger age groups. And as you can see here, we also summarize the results, and the pattern that we see is kind of similar to the one that we saw for the people 60 and above, but it's not entirely identical. We can see that for younger age groups, we see a higher decrease, a larger decrease in the rate of confirmed infection. But overall, we see that the dynamics, the time at which the booster dose effect kicks in, and the magnitude is roughly the same. In order to try and validate our approach, so as in any observational study, we have a lot of possible confounding effects and biases that could be in the data. So in order to try and address these biases as much as we can, first of all, we try to adjust for these for different covariates that we think can influence the rate of infection in the model. But in order to try and validate our approach, we also use different independent methods to try and see if we get similar results. So a different method that we used was based on matching individuals. So taking individuals that got the booster dose and trying to match them with individuals that didn't get the booster dose, but are very similar in terms of all the different parameters. So in terms of their age, the place that they live in, their demographic sector, their gender, and so and so. And what we do when we match these people, we start a kind of an experiment in which we follow these two individuals, the one that got the booster dose and the one that didn't get the booster dose. And we try to estimate what is the rate of incidence of infection in each of those pairs. And what we see is a very similar result to the result that we saw earlier, that across age groups, we see roughly a 10-fold decrease in the incidence of confirmed infection. A third method that we use to try and estimate, to validate our results, is not to compare the group of people who got the booster dose with people who didn't get the booster dose, but to compare the people that got the booster dose with the same people, with people who got the booster dose, but looking at the rate of infection during early after the vaccination, when we expect the vaccine, the booster dose, not to be effective yet. And this solves some of the possible confounding that could be between the groups. So for example, if the group that didn't get the booster dose has different behavioral attributes in terms of the healthcare seeking behavior, or the recklessness, or whatever, now we're comparing within the same group. So all of the people that we're comparing got the booster dose. And when we do that, we see that the rates that we get are slightly lower than the rates that we got in the other methods, but are still pretty high. Some of them are also close to tenfold. We can do the same type of analysis, not just on confirmed infection, but also for a severe disease. Here we compared people over the age of 60, people between the age of 40 to the age of 60, and for people below the age of 40, we didn't have enough data to produce reliable estimates. But as you can see, we see a very large decrease in the rate of severe disease, both in the elderly population and in the 40 to 60 population. As before, we can also do the same validation with the method comparing to the rate of severe disease between days 3 and 7. And we see that we get also, as before, we get lower rates, but it's still a significant protection against severe disease. And as before, we can do the same, not just for severe disease, but also for deaths. Here, we have even fewer cases, so we can only reliably do this analysis for people who are over the age of 60. And again, we see a large decrease in the incidence of death following a COVID infection. So overall, summarizing our results, we saw that we get around tenfold reduction in the rate of confirmed infection across age groups from age of 16 and above. We also see that the boosters are effective against severe COVID. For ages 16 above, we see protection that is between 6 to 20-fold reduction in the incidence rate, which means that we see around 80% decrease in the rate of severe disease. For people who are between the ages of 40 and 60, we see between 3 and 20-fold reduction, meaning that we get above 60% decrease in the rate of severe disease. And for deaths, we see also between 3 and 10-fold decrease in people above 60. So these are kind of microscopic evidence for the effectiveness of the booster. I just wanted to share with you a small amount of, let's just briefly touch upon the impact that we saw of the boosters in a more global setting, in looking at the entire country. So what you can see here is a plot of the percentage of positive tests in Israel. As you can see, and as I told you, during the middle of July, we saw a rapid increase in case numbers, which is also evident in the fraction of positive cases, positive tests, leading us to start the booster dose campaign. And as you can see, after starting this campaign, we started to see the percentage of positive tests. It stopped increasing and started actually decreasing, and we also see it in the number of new daily cases that we get. We see that the reproduction number has decreased below 1, which is in line with our predictions for the effect of the booster dose. So with that, I will conclude. If you have any questions, I would be happy to answer them.
Okay, I'd like to thank both of our speakers for their talks and open the floor to questions. I hope there'll be lots of discussion of both talks.
So if I can ask a question on the data that you just show about how many people that were vaccinated ended up contacting the disease, like the 60%, but it's 60% of the people that contacted the disease, how much that represent in actual numbers? Is 60% like 100 people and 40% also, or 100 people of like 5 million that are vaccinated, and then it's not as horrifying as we could think?
So what you're, if I understand you correctly, what you're referring to is the absolute rate difference and not the relative rate difference. So obviously, yeah, you're correct. The absolute rate difference between people who didn't get vaccinated and people who got vaccinated, even with like 2 doses, is much larger than the rate difference, the absolute rate difference between people who got vaccinated twice and people who got the booster dose. The problem with looking, so it's a completely valid way of looking at the data. The problem for us with looking at the data from this perspective is the fact that first, this rate difference is dependent on the incidence, right? So if you have like a huge outbreak, this difference would be larger, right? Mm-hmm. And the second, so for example, in Israel, what we saw is that, we get protection from the vaccines, but the spread of the disease was so significant, and we had like we had almost like 2 months of with a reproduction number around 1.3. So the disease was like growing exponentially, very steadily. And even with with everything that the vaccines did in terms of reducing the incidence of severe disease, we still would have gotten to the capacity of the healthcare system, right? So our main option was to first bolster the reduction in the rate of severe cases in people who are vaccinated, but also it's an important point to try and vaccinate as many people as we can to reduce the incidence itself. Which I think is a very it's a very important point. I think the US is very different in this sense from Israel, because you don't have like you don't have like growing incidence. The reproduction number is not very close to what it was in Israel. But for us, during the time, it was also very important to stop the spread of the virus.
Yeah. I agree with you. My comment was just that if you try people that are unvaccinated right now because they, for whatever reason, they don't want to be vaccinated, and they see these kind of numbers in percent, it's not going to convince them to get a vaccine, because they say, if you look just at those numbers out of context, sure. that was my only point.
Sure. But you're right. I think that the main thing to remember here is that we are not considering here fresh vaccines. Like we are only the second dose, like people that didn't get the booster dose are for people who got the second dose, mm-hmm. and more than 5 months have passed since the vaccination. So it's not like people who just got the second dose.
Yeah.
Other questions? So I guess I was going to ask a more follow-up question on the inhalation study, which I thought was fascinating. So you mentioned that the droplet distribution is apparently fairly similar between different size distribution was somewhat similar between different coughs from different diseases. Do you think that but what about the issue of the viral load in the cough as a function of the locus of the disease? You mentioned, of course, upper respiratory and other. You were looking primarily at deposition, but can you comment a little bit about the sources in terms of the fluid sources as when you cough, and what that might do to affect the two sides of the computation that you're doing.
Thank you very much. So as I remember, there were, yeah, that's important indeed, where the material of the cough comes. And yep, so that was, I don't remember exactly, but we checked that, yeah, one of the most common symptoms was dry cough, or it's also called non-productive cough. And in that case, we believe that the throat swab samples, the viral load of the throat swab samples are relevant, because there were viral load for two types of swab samples. Maybe the other one was nasal swab, I don't know. So we wanted to check it at that time, and maybe we could do it now more in a more sophisticated way, but we wanted to check that kind of source, what is the source of the dry cough. It's important indeed.
Yeah, so maybe I would add that in terms of respiratory droplet size generation, because your mucus is mainly 95% to 98% water, it doesn't matter really what kind of viral particles is in the mucus. When you generate the droplets, it's more based on the viscoelastic properties of the mucus. And so the original particle size is probably very similar. Now, when it dry up, the size of the viral particle itself, that may vary. But so I think it's a very reasonable assumption to use that distribution.
Thank you very much. And we also, I didn't mention it, but we also write it many times in the paper that the size distribution is valid very close to the coughing person. So if it's further in time or space, then the size distribution will be very different. And of course, probably the viral content on the concentration will also change, and it's not homogeneously changed, so it's difficult then.
Okay, well, when your viral load estimates in the fluid differ by a factor of 10 to the 8th, that's a pretty big range.
Yep.
So you used the geometric mean, which seemed to make sense, but that's a hard one to deal with, I suspect, if you want to actually calculate a number for infectivity.
Yep, of course, but even if you use the maximum number, then it was still very, very low.
Okay, other questions for our speakers? Well, I'll ask one on the epidemiology. I know it's very, very early, but do you have any sense of what you're seeing in terms of your curves? Your curves look pretty flat. So does that suggest that the third dose is conferring longer term immunity than the first two shots?
Yeah, that's the $1 million question. We're looking into it. We don't have robust data yet. We're actually we are coming up with methods for monitoring it better. For example, one interesting way of monitoring it, it's usually very difficult to do these kinds of monitors, because when the incidence goes down, your statistical power also goes down, and then it becomes difficult. One thing that we thought about doing is in Israel, when people get into the country from abroad, they all get tested. It's like 96, 97% of the people get tested. And then if we have a high incidence or high prevalence of disease in other countries, we can try and use this fact with the fact that people are getting tested to probe the efficacy of the booster throughout time. So this is one of the sensors that we're probably going to set up to track the whether the boosted immunity wanes or not.
I mean, a follow-on question, you talked about doing a sort of internal control versus external control to look for behavioral differences. And is there any, are you seeing people being more relaxed after the third booster shot? I'm not entirely guilt-free here, because I would not have gone on this trip if I hadn't been able to get a booster shot in advance. And so here I am taking more risks because I had that third shot. Of course, that would make your number, if people were doing that, then your actual improvement would be better than it looks from the numbers.
Exactly.
But
yeah, so in theory, it should lead to a more conservative underestimate of the effect. It's true. I think, it's very, very difficult to get good estimates of the behavioral changes. And actually, I think also in a sense, the thing that you're really interested in, it includes the behavior changes, right? Because like what you're interested in the end is when you vaccinate people, what will happen to incidents. So it's kind of it's a tricky question. I think in most cases it's very hard to tackle it. What we can see is that people are actually not getting tested for several days after getting vaccinated. You have all these different kinds of biases. You have a bias that's called the healthy vaccinee bias, which basically means that people who are feeling sick, they don't get vaccinated. And then only you get like at the beginning, you see that the boosters are kind of effective, but it's only because of the fact it's like a transient bias that you see at the beginning. And after several days, it should disappear. So we have all these different kind of behavioral biases. We're trying our best to use as many independent and methods that all have different caveats, but combining them, we hope that we get a kind of a robust picture of the situation.
Other questions? Our panel seems unusually quiet today. Lorenzo, do you have anything to add?
Ooh, not really. It's not really my field. I have some difficulties making questions on that area.
So maybe coming back to Balazs, if we were going to be trying to do experiments to try to clarify the role, the role of particulates, what would the experimentalists need to do that would be most useful to you in improving your understanding of deposition?
Thank you very much. What we are thinking now, that based on this research, it seems to be a very, very important question how virion, by the way, any other infectious particles, bacteria, other things reach the deeper airways from the upper respiratory tract. And it's not an easy question because it's difficult to measure anything inside the body, but we will be interested in what is generated during inhalation in the upper airways, and yeah, that will be a very interesting question. And there is another part which will be interesting, and it's not really physics, but what we are thinking is whether we can catch any steps between the upper airway infection and pneumonia. And for example, if we could see some early so infected spots in the deeper airways, in the interthoracalis airways, that could be interesting. And what was shortly discussed or mentioned during our working group aerosol transport, that maybe deposition hotspots can play an important role, because we see this very low deposition probabilities. And if we suppose that not only one virion, but 2 or more is needed to infect a single cell, then it can be very important where there are enough virions and enough receptors. And Chantal also mentioned, and I tried to check the literature, that there is some, ciliated cells are more frequent in the peak of the bifurcations, and they are one of the most important which express this ACE2 receptor. So that is, if I had the opportunity, I would look at the carina regions in early phases of the disease, whether we see any clue that it goes to that way. Yeah, thank you.
I mean, you mentioned all the whole list of quite complex mechanisms for transport within the respiratory system. The airway, the reinhalation, transported fluid, and so on. So I'm sure that some of those are probably better understood than others.
Better understood than, sorry?
Well, for example, mucosal transport, I suspect, is pretty complex. You mentioned diffusive transport, basically local diffusion of virus within a tissue, transport in mucosal fluid, reinhalation, and so you have a lot of processes, as you say, between moving the virus around within the body. And again, I guess one question just for people to think about is which of those need the most work, which are the least understood of those processes?
Yeah, I think the mucociliary transport is very hard, not only to measure, but also to model because of the viscoelasticity properties. And that's extreme. There is no real good models out there. I mean, there are attempts, but I think that's one of the least understood, or the worst, not worst may be a bad word for people that do that kind of modeling, but the more challenging modeling endeavor is this mucus transport and how they break with shear forces to generate these droplets. The modeling of generation of respiratory droplets is basically inexistent. What's there, it's based on simpler law because that's the best we can do at this time. So anything that informed that from an experimental value, maybe in cell culture where we create shear stress, and I don't know, that would probably be of high value.
Okay. Other questions? No. Yes. I see John is off. Any of our other members? No, well, thank you then. I know it's late for people. So I want to thank our speakers very much for the 2 talks. They were very timely and appreciated. And
Thank you.
I appreciate you taking the time to join us today very much.